Okay, hi, my name is Mark Pepin. I'm a current Humboldt Fellow working here at the Institute for Experimental Cardiology at the Heidelberg University Hospital, working in the laboratory of Dr. Johannes Box. And we're standing right outside of the building where the lab is located. Just wanted to point out to you here, this is in Heidelberg. And the lab itself is actually on the second floor. You should be able to see it right outside. But we'll take a look inside and see what we find. Okay. The entrance of the building, so you can see here. At this moment in time, we are all wearing masks in the building, surgical mask or an N95, whatever you prefer. But we'll put it on and then go upstairs. Okay, so we made it to the second floor. First test of endurance is walking up 2 floors with the mask on, but we did it. So I wanted to first introduce you to the lab itself in picture form. If you could walk over here with me. So this is the lab of people without masks, because I've actually met very few of them without them. But this is the lab. And the building itself is divided into 2 parts, 2 halves really. One side is the administrative space. It's where the DZKK management is located, as well as Johannes Box office. But then on the other side, behind you, in front of me, is all of the laboratory space, where all the benches are located, cell culture, and a variety of other tools that I'll show you in a minute. Marco, hey.
Look at you, okay. Yeah, hi everybody, my name is Marco Hagenmüller. I'm working in the Box lab since 6 years now, or 5.5 years now. I'm responsible for all the stuff that nobody wants to do, especially writing protocols for animal experiments, making figures for the publications, being responsible for all the biosafety level 2 stuff. So there's always a lot of things to do, and nobody wants to do it. I am the one who does it.
Okay, now the next office we're in front of is that of Dr. Johannes Box and the administrative assistant Sylvia Harak. And hey, hello. Sylvia is here. Obviously busy as always, but without her I'd probably still be stuck in customs. So we have a lot to thank for her kind of facilitating transitions of different postdocs and other personnel in the lab, and also getting us paid, which is helpful. Okay, so let's move on. Okay, so now we're about to enter the research space with all the benches and stuff. So here we're standing in actually the locker room with a bunch of lockers here for people in the lab to store personal items, and also lab coats here, which we'll put on before we enter the bench space, and then we'll take a look inside. Come with me. Okay, so we're now in the Box lab, which is basically this entire area behind you, and then to the end of this wall here. So the first person you should always talk to if you enter the Box lab is this person right here. So this is Jutta. She's really the manager of the space. She's coordinating all the technicians. And if you don't know where anything is in the lab, you should always ask Jutta first, right? Right. Okay. And she will always tell you if you're doing something incorrectly, which is super helpful when you're starting out. So, thanks. Okay, and then over here, this is Matthias. Hi, Matthias.
Hello.
Would you mind telling us what it is that you're doing?
Yes. So I'm preparing samples for a Western blot, and this is basically related to something that Johannes presents in his talk. So he will probably present that we developed compounds that inhibit the protein-protein interaction, and I'm currently testing these compounds in cardiomyocytes to see what they do regarding gene expression, phosphorylation, and so on.
That's awesome.
Yeah.
Thanks for explaining. Appreciate it. Okay, let's move on and I'll just show you a couple of other things down the lab. Okay, so this is also the room right in the middle of all the labs where the computers are located. If you have echocardiography data that you need analyzed, there are computers in this room that you can do that. Also, histology can be analyzed in the same room. Okay, so this is the end of the Box lab. What I wanted to do real quick is show you the Confocal Microscope. It's a really cool technical resource that's here in the Institute, but it's upstairs and we're gonna go take a look, see if anything's happening there. Hello?
Hello.
Hi, Sumin. do you have a moment to explain what you're doing?
Yeah, sure.
Okay, great. Let me give you the microphone. Okay, so what are we looking at here?
In our lab, we feel very interested in studying the function of lipid droplets in the epigenetic regulation of the cardiac function. And with this aim, we developed several in vivo and in vitro models. Will help us study the function of cardiac metabolism in the cardiac function.
Fantastic. Thank you so much.
You're welcome.
All right, I'll see you later. Okay, now we're going to take a look at the Ips Cell Culture. And here we have Aicha.
Hi, I'm working with the inducible pluripotent stem cells and differentiating them into cardiomyocytes, which are the heart cells, as you might know. And our aim is to reduce the number of animals used in the field by using these cells from humans.
What kinds of tissues are you using to make it?
Our original tissue is the human blood, and from these we take the stem cells and reprogram them to be inducible pluripotent stem cells, and after that we go through the differentiation protocol. Once we produce the cardiomyocytes, we can test possible drugs or chemicals for different diseases, different heart diseases, for example.
Fantastic.
Thank
Wow,
you.
well, thank you so much for your time.
Thank you.
Okay, so now we're down here in the basement in the mouse facility in the S2 culture room. And this is where I'll show you some of the mouse phenotypes that are being used as models of heart failure in the Box lab. So let's go take a look. Here we're at the echo room, so we'll see what's going on here. Matthias, hey, do you have a moment?
Yes.
Okay, hi Sabina. Okay, would you mind just explaining to us what it is that you're doing?
Sure. So here Sabina is doing echocardiography of mice, and what you see here on the screen basically is a normal heart. So you see the left ventricle and you see the heart pumping blood, and this is how a healthy heart in the mouse looks like. Sabine, maybe you can show us an image of a heart failure heart. Yes, so what you see here, this is a heart failure model that we use. It's called TAG surgery. And this is a typical model that we use to validate the mechanisms that we find upstairs. So we always want to translate the basic mechanisms that we have from our biological, biochemical experiments into in vivo relevance, and eventually then also test compounds in mice that we developed.
Okay, well that concludes our short lab visit. We didn't get to see everything, but I hope that gives you a little feel for what it's like to work in the Box laboratory. For any additional questions though, please do not hesitate to reach out through email to Dr. Box or myself. We'd be happy to chat further about what it's like to work here and the future projects that might be in store. So thanks, and I'll see you later.